Hello, it's Jim, the author behind the blog Reverse Engineering the Origins of SARS-CoV-2 in the book COVID-19 Mystery Solved. This is going to be a video about the origins of my blog and my book because I get the question a lot. Who are you? And where did you come from? To give you some background, this book was brewing in the background for 3 years. I'm recording this in 2025, but most of this book was debated in 2022. As I write here, the above blog post originated from a March 2022 Twitter thread, which asked the simple question, why did Xi Li of the Wuhan Institute of Virology publish RATG13? The answer is not easy. It came out during the drafting of the proximal origin paper, but my book provides one. That February 2022 question led to a debate with Rutgers professor Richard Ebright. He argued that Xi Li could have engineered a genome like SARS-CoV-2. I countered this was implausible, citing Ralph Barrick's own statements on the technical challenges involved. To give you a background on the importance of RATG13, it's on the cover of my book. This is the genome here at the top, and this is the SARS-2 genome at the bottom. It's a 100% match. It's called an alignment. It's what virologists do when they look at a new genome, and it wraps around to the back. RATG13 on the top, SARS-CoV-2 on the bottom, and it's pretty obvious when you do the alignment. What's the difference? Is this S1, S2 out of frame insertion of what's called the infamous furin cleavage site, PRRAR. So it's very easy to see if you do what's called the alignment. And Dr. Fauci was told by Christian Andersen, we'll talk about him, that we find the genome inconsistent with expectations from evolutionary theory. That was on February 1st, 2020. Fauci later replied after the teleconference, I see it. So when you do the alignment of the genome of RATG13 over SARS-CoV-2, it becomes very easy to see that this genome called SARS-CoV-2 was engineered. So I'll start off by giving you a quick little background on Xi Li. She's a relatively well-known bat virologist inside the small bat virology community, but she wasn't well known outside of it. She's probably the premier bat virologist in the Wuhan area, if not all of China. So she entered into the international limelight on January 23rd, 2020, the same day the Chinese authorities sealed off Wuhan to contain the new SARS-2 disease. In a preprint paper, her team announced she had found a virus 96.2% similar to SARS-2, and that one was called RATG13. When she published it on January 24th of 2020, a lot of attention was on the wet market or even the BSL-2. Wet market, if you're natural origin, BSL-2, ironically, about 300 yards, 1,000 feet away from the wet market is a BSL-2 lab. It was just opening, so it's no longer a suspect. It wasn't an operation. But at the time, people sometimes suspected it being the origin. But when she published RATG13, she brought attention away from that lab and even away from the wet market and towards her lab, which is across the Yangtze River. On the south side of it is her BSL-2, which is closer to the center of the downtown area, Wuchang to be particular. And about 20 miles south of her lab, BSL-2 lab, is Daniel Anderson's BSL-4. So I give you a quick background on my book. I literally bring her up at the beginning. So I base it on the information of two whistleblowers, I say here. One from within inside the Wuhan Institute of Virology and the other from the U.S. military. That was Xi Li and Major Murphy of the DARPA fused proposal. Though their identities are known, their stories remain untold until now. I go on to say the lab leak narrative you heard is incorrect. A $100,000 bet was lost because until now there was no biological evidence for Wuhan lab leak. SARS-CoV-2 does not infect humanized mice or Chinese bats, but transmits well in American lab bats. Instead of debating the lab leak, we will focus on what leaked. Lab leaks happen daily, but only infect the technician. We will explore the origin of the virus and expose its creators. SARS-CoV-2 was designed to spread. Contagion was a feature, not a bug, until the animal vaccine jumped species in the Wuhan Institute of Virology. International scientists audited the Wuhan lab on January 31st, 2020. They passed, but others failed. So that's what I'm referencing in the back of the book right here. This is the audit, where Fauci's been told that the genome appears to be inconsistent with expectations from evolutionary theory. So that's a lot to unpack, but I'll start off with my own story and when I got interested in the lab leak debate. It was relatively late. I thought it was kind of over, to be honest with you. Lab leak had basically been proven. So I always got more interested in wet leak. But my own interest in lab leak was peaked here in January 2021. So that's about a year after all this happened. I thought most of the evidence, I always assumed lab leak, but I thought most of it would just be covered up. But this paper by, not more than a paper, it's a long, essay by Nicholson Baker in New York Magazine was quite the eye-opener. Not because of all the mentions of Xi Li, which is about, 15 times. It's the number of times he mentioned Ralph Barrick. Nicholson Baker had written a lab leak book right before this COVID outbreak occurred. So he was hit the ground running, and he did some deep research on this. And to name Ralph Barrick, this white guy in North Carolina, 40 times was just amazing to me. That was always like just complete interest, which is more than, twice as much as Shizing Li. And it was twice as much as the character we all know, Peter Daszak. He was only mentioned 23 times. So then Fauci's, of course, mentioned quite a few times too. So about 80 times for all three, guys, compared to just about 15, 20 times for Shizing Li. So there was always a bit of a cloud around Ralph Barrick and Peter Daszak, but it wasn't, they weren't a primary suspect because it's Wuhan. It's not North Carolina or New York, which is where Ralph Barrick and Peter Daszak are based. So when I got involved in Lab Leak, I was very friendly with the Lab Leakers, as we'll call them, Richard Ebright, Jamie Metzel, kind of stuck here in the middle, asking a funnier question I thought would be who Shizing Li was paid for, or who paid Shizing Li more, the PLA or the Pentagon. Because even after reading Nicholson Baker's piece, I still assume it has to do with Shizing Li's lab, and Shizing Li may have covered this thing up. And Richard Ebright replied, PLA got more for its money. But then, I did an interview recently. I'm recording this in 2025, and Jamie Metzel picked up on what I've been called by others a second-generation lab leaker, because I came along much later than they did, and I got interested much later than they did. I came with a fresher perspective than they did. So that's been my outlook. And along came Richard Ebright to now sour on me and call me ignorant as shit for brains. So to fill in the blank between 2021 and 2025, it starts off mainly in 2021. So this is September of 2021. The DARPA Diffuse grant has been leaked. We've talked about it in other videos. And inside this grant by Ralph Barrick, Peter Daszak, Shizing Li, and Linfa Wang, who's being interviewed here, or part of the interview, there's a mention of a fear and cleavage site. So this is now, September 30th, about a week after DARPA Diffuse is leaked, and we don't know who wrote that piece of the grant. It's a large grant, 75 pages, and inside of it was a couple of lines saying we will insert a fear and cleavage site into a novel coronavirus. Just to give you a quick background on that, how odd that is. That is about as odd as it gets. This is it's the smoking gun for genetic engineering. So it's whenever you see anyone talk about it, it's what you start off with. That's the fear and cleavage site, PRRAR, in the SARS-2 genome, and it's the only fear and cleavage site found in a sarbecovirus. This is a list of 2 or 3 dozen sarbecoviruses. None of them have a fear and cleavage site. The S1S2 border, but the SARS-2 genome does. That's why we still talk about SARS-2 genome, 5 years after it emerged. So to give you a quick background, DARPA Diffuse is leaked. This is Alina Chan. She asked a pretty tough question to Linfa Wang, who was a co-PI in the DARPA Diffuse proposal. And listen.
In fact, we have no insight to all the viruses collected at the WRV after 2016. If you read the literature, all those viruses have been collected before 2016. So we have no idea what they've found. And if you read the DARPA proposal, they said precisely they were copying parts from nature. They were looking at natural furin cleavage sites and putting these into SARS viruses in the lab with slight tweaks. So what we're saying is completely consistent with a lab escape of a virus that had a furin cleavage site from nature put into it. So my question for Linfa is, who proposed this furin cleavage site insertion in the paper? Was it Shizing Li? Did she propose to put the furin cleavage site? So was it?
No, that's not. I don't know who proposed. That's not the part. We each have a part, right? The Wuhan Institute was the field research. I the best immunology, and UNC is doing the virology. So that it's pretty obvious, right? Yeah.
No, but who proposed the idea? Because the idea says that we are reviewing all the deep sequencing data. We've seen these cleavage sites, and we're going to put them into SARS viruses in the lab. So the question is, who had the idea and access to this data and could do it in the lab in a seamless way?
UNC, right? It's a UNC lab. The grant is very clearly.
It's University of North Carolina.
Yeah.
So the answer is pretty obvious. Linfa Wang, basically an insider, has now told us outsiders that it was Ralph Barrick's idea to insert the fear and cleavage site. So along came Richard Ebright.
Very normal. In fact,
he was pretty blunt with his answer. His first answer was, I don't know who proposed that. His second answer was UNC. Either the first answer was a lie, the second answer was a lie, or both answers were lies. In this view, the fact that his answers to other questions were demonstrably untruthful is hard to choose. He's basically called Linfa Wang a liar for telling us that Ralph Barrick proposed to insert the fear and cleavage site. Fast forward 4 years. 4 years, basically. 3 years, to be exact. 3 years and change. Ralph Barrick went totally quiet and never talked to the media ever again. He was talking to Lab Leak reporters. He even talked in this piece to Nicholson Baker. Nicholson Baker got him on the phone. But Ralph Barrick went quiet for about 3 years, and he finally emerged to say, yeah, it was my idea. And he never really came up with an alibi either. So this is, to give you an idea, this is when I first realized maybe this cover-up is bigger than just Wuhan. Maybe it's bigger than just virologists, maybe even the pro-lab leakers are in the cover-up now. I don't want to get into why, draw your own conclusions, but, it changes the dynamic, quite a bit if COVID-19 was developed on U.S. soil and tested on Chinese soil. So even Alina Chan later on, said, according to Linfa Wang, Barrick was the one who came up with the idea of putting the furin cleavage site on the Sarbeca virus, like SARS-like virus. And she and Wang are both very highly respectable virologists internationally. Here came Richard Ebright again to protect his boy Ralph Barrick. Xi Zhengli and Linfa Wang are strictly mediocrities that were largely unknown before the pandemic. They were both very well known. Both of them. Linfa Wang's in the New York Times before the pandemic, and Xi Zhengli is very well known within the bat industry. Xi and Wang, each just first Arthur, senior Arthur, he goes on to call them, sub-mediocrities, essentially. So, it's a bit of a narrative that's formed, around this DARPA defuse proposal, which had me rethink everything. So then after Linfa Wang threw Ralph Barrick, under the bus, and Barrick didn't defend himself, about 3 or 4 months later, Peter Daszak came out and said the same. It was Ralph Barrick's idea. And I said, did you ever warn about Ralph Barrick? And then, did you quiet on this Daszak interview? Was my point to Richard Ebright. Daszak's interview responses are a cesspool of fraud. Daszak and his equal never funded. See, he changes the subject when he doesn't like the question. He wants to talk about humanized mice, I think, doesn't he? Yeah, humanized mice. SARS-CoV-2 doesn't affect humanized mice, by the way. To claim it infects humanized mice is to claim it affects raccoon dogs. We've talked about this. It's the importance of the SARS-2 furin cleavage site. But what we haven't talked about is what started my my whole endeavor down this road. And it was this March 2022 Twitter thread. This is it, March 2022 Twitter thread. So now DARPA defuse is leaked. Both Peter Daszak and Linfa Wang have ID'd Ralph Barrick as the gunman, the one that inserted the furin cleavage site. And Ralph Barrick's not defending himself. So I'm basically hit the rewind button on the entire lab leak narrative. And this is targeted at. It should have been here's Richard Ebright, here's Alina Chan. And this probably should have been Matt Ridley, the co-author of the book on Alina Chan, this three-headed monster of misinformation, as I call it, looking back upon it. I mean, it's easy for me to come along, with hindsight 2020 after DARPA defuse leak, but I'm at least, I pivoted, and I think we should all pivot after some shocking document like that emerged. But a lot of the evidence that they had was it was conspiratorial evidence, meaning Xi Zhengli didn't even, declare the furin cleavage site in the SARS-2 genome. And you'll hear that a lot from lab leakers. And actually, Xi Zhengli did. Here she is in January 31st, 2020, with the two co-authors based in, the Shanghai area, I believe. We predicted that the cleavage site for generating S1, S2 subunits located at 694, 695. They're off by, a few nucleotide in the grand scheme of things. That's how bad their genomic engineering can be at times. Here's Xi Zhengli in March of 2020. Here she actually aligns it correctly, 685, 686 at the S1, S2 border. So Xi Zhengli did declare the furin cleavage site early in the pandemic. And it's easy, hindsight's 2020 to see it now, but we've all had time to recognize it. But there's another person that did not declare the furin cleavage site early in the pandemic, and that was Ralph Barrick. He did not declare it here in this paper that he published early on in the pandemic, January 28th to be exact. So he did not declare the cleavage site at all. He talked about the receptor binding domain, which is what they talked about a lot in the proximal origin paper. So Ralph Barrick's colleagues, I don't think either talked about it. So these are two of his former postdocs, in early January 2020, no cleavage site declaration. So what happened when Xi Zhengli published RATG 13 on January 24, 2020? It became easy to see the SARS-2 furin cleavage site. It's called the alignment, and it's a 100% amino acid match, 250 to the left, 250 amino acids to the right, and right in the middle of the spike at this S1, S2 border is this furin cleavage site, PRRAR. So when Xi Zhengli uploaded this paper and genome on January 23rd of 2020, Christian Andersen went from talking about snakes and natural origins on the Virological Forum to contacting Eddie Holmes and Eddie's reaction when he saw the alignment. This is now Christian Andersen at the Scripps Institute has downloaded Xi Zhengli's RATG13 genome early in the pandemic, and he's overlaid it with the SARS-2 genome, and he's seeing what we're seeing. And this is what Eddie's reaction was. Fuck, this is bad. So it's scripted. It's documented here in Jeremy Farrar's book, Spike. And there's also an email here from Bob Gary. He's a colleague of Christian Andersen. Bob Gary is based in Tulane, and he says, I really can't think of a plausible natural scenario where you get from the bat virus to one very similar to it, where you insert 4 amino acids, 12 nucleotides, all to be added at the exact same time to gain this function. And you don't change any other amino acid in S2? I just can't figure out how this gets accomplished in nature. do the alignment of the spikes at the amino acid level. It's stunning. Of course, this would be easy in the lab. So this is what Bob Gary's writing about right here, the alignment. So no one really suspected engineering a SARS-2 from January 10th to January 24th. January 10th is when SARS-2 was published. January 24th is when the RA2G13 genome's uploaded. And evolutionary virologists who have the software to do the alignment and know how to do it very quickly and easily all now think it's engineered, but only after Xizeng Li has uploaded RA2G13. And that kick-started such a cover-up. Here's Fauci, in the middle of, Saturday morning, Friday night, talking about a Barrick-Shih paper. We'll talk about that in a minute. So these are one of many myths that you come across. And that Barrick-Shih paper, Fauci was up until midnight. He was suspicious of it, as were many others, because it has Ralph Barrick and Xizeng Li, on it. This is a 2015 paper where they created a novel chimera. And it was so suspicious. There's actually an editor's note here on the Nature magazine. There is no evidence that this is true, that this paper is being used for unverified theories and conspiracies that the SARS-2 genome is engineered. So we found out why Xizeng Li was a co-author on Barrick's paper. Is because Xizeng Li provided the genome that they used to experiment with, called HHC014. And Ralph Barrick's 30-year bench scientist, Boyd Yount, designed the infectious clone and recovered the chimeric virus. He's the specialist inside Barrick's lab in North Carolina, and Xizeng Li just provided the sequences. And Barrick went on to explain why. And Xizeng Li was gracious enough. This is Barrick being interviewed before DARPA Diffuse leaked. He was talking to the media after DARPA Diffuse leaked in September 2021. He quit talking to the media. But before it leaked, there was a lot of suspicion around Xizeng Li, and basically the suspicion is based upon the idea that someone like Ralph Barrick taught Xizeng Li how to engineer something like SARS-2. And Barrick has said, no, I didn't, for various reasons we'll get into. One was that he never uploaded the genome sequence to show the Chinese how to do it. But in other words, though, Xizeng Li, this is Ralph Barrick talking, Xizeng Li was gracious enough to send us, University of North Carolina, those sequences almost immediately. In fact, before she'd published. That was her major contribution to the 2015 paper. And when a colleague, he calls her a colleague, when a colleague gives you sequences beforehand, co-authorship on the paper is appropriate. He goes on to continue here. This was the basis of our collaboration. We never provided the chimeric sequences, clones, or viruses to researchers at the WIV. And neither she nor any members of her team ever worked with our lab. No one from my group has worked in WIV laboratories. So Barrett calls her a colleague, basically, because she shares genome sequences with them. But he does not train her or work with her, per se. So their technology is so different. They're not even really competing, nor are they, really truly collaborating. They just share what they can't even work on. Xizeng Li couldn't work on the SHC014 genome. She couldn't isolate it. So she sent it to Barrick to create it, and he was able to create it upon an email. She just has to email him the genome sequence, and he could download from there and create a novel chimera based on an email sequence. So here's another example of Xizeng Li not being included as a co-author on a follow-up paper in 2016 called WIV1. This is basically the SARS-1 genome. And even though Xizeng Li sent him the plasmids in 2016, she's not included because it was already published, the WIV1 genome, as opposed to SHC014, which was not published, so she was included as a co-author. So I'm trying to separate here the fact from fiction and kind of the gray area between the two, as they, they work together in a kind of a funny little way. It's more of a division of labor than it is a collaboration. He does what he does in the lab, and she does what she does in the field, collect bat sequences. So, yeah, I go on to say this myth is important because it leads to a bigger myth that she and the Wuhan Institute of Virology had a reverse genetic system to create SARS-CoV-2. So guess who perpetuated that myth? It's basically Richard Ebright, to be honest with you. It's kind of behind most of the myths and a lot of this threading. So whenever he references a paper, he's referencing the bacteria artificial chromosome. And that is essentially a used Yugo and compared to Ralph Barrick's cDNA system. So it's we'll get into that a little bit more here. Christian Drosten helped me sort it out a little bit. So Christian Drosten has said, in this podcast, this is basically he's been called the Fauci of Germany. It's more appropriate to call him the Ralph Barrick of Germany. So he's basically gone on to say, look, I actually believe to this day that a lot of this novel chimera work was done in the USA, and those viruses are then sent to China for certain experiments. So he's saying only the source of the virus genome in China is on the spot. But for certain experiments, these experiments are actually carried out in the USA. So he's trying to sort this out too. And Ralph Barrick has come out and said, Xizeng Li, she has what's called a WIV1 molecular clone. And based on that backbone of the WIV1, SARS-1, they shuffle spikes into and out of the backbone. So they can shuffle spikes, but they can't create new backbones like SARS-CoV-2. And the WIV1 genome is about 20% different than SARS-CoV-2. Christian Drosten went on here to say, this is again my March 2022 Twitter thread trying to punch through a bunch of myths. And he's talking about here about the Vero cells, I believe, that would delete the furin cleavage site. And he's talking about her reverse genetic system that doesn't even has the ability to create a new genome. And he goes on to give a great analogy, about a new car radio. The Wuhan Institute of Virology can create a car stereo, but they can't create a new car. Is basically what he's saying here, which is what SARS-CoV-2 is, a brand new car. So Drosten, who's basically, again, the Barrick of Germany, has had a keen interest in DARPA diffuse because he understands what the furin cleavage site was to be done in the University of North Carolina. So along with the idea that Xi Jinping covered up the SARS-2 furin cleavage site is actually someone that did cover it up. And his name was Ralph Barrick. And he did here, this is a February 2020 presentation. This is Ralph Barrick giving a presentation to congressional staff in February 2020. None of them are wearing masks, but it is after the pandemic has started in Wuhan. So Ralph Barrick's been called, ironically, as the coronavirus expert in the U.S. government to give a presentation on this new genome that's emerged in Wuhan. So Ralph Barrick, DARPA diffuse hasn't leaked, but obviously he knew about it because he wrote the grant about the furin cleavage site. So he knows about the idea of the furin cleavage site in the SARS-2 genome. So he doesn't declare it, obviously, here in this presentation. Again, this is congressional staff sitting in the basement of the Capitol building in February 2020. So he's going to do a slide comparison of SARS-2 to SARS-1. And he should, at this point in this 1 hour long technical presentation, declare the furin cleavage site. But Ralph Barrick doesn't. It's a lie by omission.
The genome of the new coronavirus looks just like SARS. Brown is replication genes. Green are structural genes. Gray are genes that regulate virulence. But there's a lot of variation in them, so we don't really know what they do.
Awkward sigh there. He knows what he should be doing. He should be declaring the SARS-2 furin cleavage site. So SARS-2 is up here, SARS-1's down here. That's the only difference between the two. And he doesn't say it at all.
This is the receptor virus.
He goes on and talks about the receptor binding domain. So guess who did mention the furin cleavage site? This is a little bit later in 2020.
4 peptides in the S protein. 3 of them are located in S1, and 1 is a furin cleavage site located in between S1 and S2.
So Xi Jinping did declare it. This is later on in 2020. There's another one where she's declaring it in Chinese and Mandarin in early 2020. So after DARPA diffuse leaked, Barrick went quiet. So here, Vanity Fair, no response. Even to his own, Nature magazine, he's not responding. That's a scientific journal that's obviously, on the natural origin side. This is the Telegraph, not the Telegraph. This is the Intercept. Barrick did not respond. And even Science magazine here, this is a funny one. The application which Dazek and others submitted to DARPA outlined a variety of research projects. One experiment to be done by Ralph Barrick would make the human enzyme to cleave their viral surface proteins, that's the furin cleavage site, and increase the infectiousness and thus the pandemic potential. Barrick did not respond. So Barrick's not responding. He's gone radio silent and after DARPA diffuse is leaked. I pivoted. I've now got Barrick in my sights as the probable culprit behind this, but everyone else is kind of, stuck in their own narrative. Sunk cost is probably the better way to put it. So here's my the second part of the right here, the second part of the paragraph, I should say, the second paragraph. The February 2022 question led to a debate about why, or it's actually the first part of the thing, which is essentially why did Xi Jinping publish RATG13? So I'm going on to ask this question now to, I believe, Richard Ebright in February. Richard, why do you think Xi Jinping published RATG13? He snapped back with a quick answer, like with everything. To improve chances of acceptance of a paper in Nature. That's the exclusive scientific paper for virologists and scientists. And thereby qualifying for 140,000 yuan, or $20,000 US. I don't know where he got this. It's thinly sourced at a Chinese paper that Chinese scientists would get a bonus if they published in Nature. And even if she did get the money, about 10 of that 1,000 or 20,000, that's a lot of money to be published in Nature. You have to pay Nature, thousands of dollars in order to get published in Nature magazine. And so I say best answer yet, after thinking about it for about 5 seconds, but $20,000 for a lifetime of questions and career suicide. Christian Anderson and Bob Gary don't email Fauci until after she published RATG13 on January 23rd, 2020. She made that furin cleavage site stick out like a sore thumb. And I, unlike him, give my references. Virological.org is the forum that the virologists were hanging out on early on in the pandemic. And once Xi Jinping uploaded RATG13, the forum went quiet. You can literally see them like vacate the forum for pretty much the entire month of February as they went underground to their Slack, into their emails, into their burner phones to talk about this thing being engineered. So they quickly pivoted from a public stance of it being perfectly natural, and they perfectly believed that. But once they had the RATG13 genome, and he says right here, this is Bob Gary's buddy, Bob Gallagher in New Orleans, the publication of a highly similar RATG13 sequence about a week ago has fueled this type of speculation. At first glance, the alignment of the sequence of both is natural, but on the either side of this new furin cleavage site, the amino acid sequence is identical. It's 500 amino acids in total, so it's a 100% match on either side of this SARS-2 furin cleavage site. So that's essentially what's happened here, is this genome now went from looking natural to unnatural. So RATG13, again, was not in the public domain before the pandemic. Xi Jinping brought it out after lab leak, and ironically, that helped make her, the primary suspect of a lab leak. I don't even know if we talk about Xi Jinping if she doesn't upload RATG13. I'm sure we do, but not as much as we do. But by the fact that she uploaded it, it actually brought attention to her lab. And so it makes it a difficult question to answer. But once you understand who inserted the furin cleavage site, when she published RATG13, she said, "Not my lab." She said that very proudly. But when you understand Ralph Baric inserted the furin cleavage site, a lot more is going to make sense. So it's just keep this in mind that genomes are shared across time and space in the mail. They basically, Xi Jinping emailed Ralph Baric, these SHC014 and WIB1 samples. So Baric could, theoretically do the same. To make a long story short, just go here to post 6, which evolved into chapter 15, and I'm being pretty honest about this. If you believe the Wuhan Institute of Virology engineered SARS-2, then published RATG13, you are a simpleton, unlike Christian Anderson. So I hate to be like that, but that's what you are. You're a simple-minded person, because this is a complicated subject. This guy right here is a world-class evolutionary virologist with a world-class view of the Pacific Ocean out of his San Diego office in California at the Scripps Institute. It's what he does. He studies the evolution of genomes, and he's got this fancy, software he can do it with. So I read this and just get your mind around it. And he goes from thinking, the genome is natural to unnatural. The minute Xi Jinping uploaded RATG13 to GIS-Aid, she had to upload it to GIS-Aid because there was an embargo. She had partially uploaded the genome in 2018 to NCBI, but it wasn't published until a 4-year embargo in 2022. So Xi Jinping went around the embargo and uploaded to the competitor, GIS-Aid. Xi Jinping, January 24, 2020. So a fecal swab is what it was from a Rhinophilus affini bat. That's a Chinese horseshoe bat, which is the source of SARS-1, and you'll find in the DARPA Diffuse grant as being a pretty important species to test genomes and viruses on. So this is a long post, so go through it about the burner phone and the emails and everything else that's involved in it. And I think at the very bottom, it's pretty obvious why Xi Jinping published it, because Ralph Baric wanted her arrested for publishing it. It kind of made his own work kind of stick out like a sore thumb. So it's not hard to understand if you just come at it objectively. So other people have asked why Xi Jinping even published the RATG13 genome. This is Eddie Holmes. Wondered, why does Xi's group even publish it? That would only help point the finger at them. It makes no sense. Well, she's pointing the finger at Ralph Baric, but none of us knew until now. And so Ralph Baric went on to say, she should be arrested, I believe, down here at the very bottom. And it's a bit of an inside, story, but it's a good one because it's got a bit of a plot twist to all this. So it's an interesting one. There's a lot of emails to go through here. You can pretty much avoid most of them, but here's the Slack message where Barrick's sitting in Fauci's office and he's talking about Xi being arrested right down here at the bottom. The Chinese government, and Barrick said that Xi Jinping's paper was not approved by the Chinese government. She could be arrested for it. So Xi Jinping has become the whistleblower. I have turned her into, in essence, the hero. In the back of the book, I talk about her being, 2 whistleblowers, and she's one of them. So there's supposed to 2 whistleblowers, one from inside the Wuhan Institute of Virology, and that's her. So other people have been asked, why did Xi Jinping publish RATG13? And here's Alina Chan's guess. And it's kind of a convoluted, long thread here. You can go through it if you want. But at the bottom, she basically says it was a race for recognition. And she kind of brings in this conspiracy of trying to cover up the 4991 genome. The 4991 genome is the old name of the SARS, or the RATG13 genome. That's what it was previously called. It was renamed RATG13 in 2018. So that's another conspiracy that came up. It was renamed after the lab leak. No, it was renamed RATG13 before the lab leak. But in 2013, it was collected from this Mojang mine shaft, which was connected to 3 dead miners. And that alone was another side conspiracy that kind of comes to a dead end. But out of this sample, they collected this 370 base pair nucleotide fragment of the RDRP, and it was labeled 4991. And it's a 98% match of SARS-2 over a very small segment, 1% of the genome. But then when Xi Zhengli published RATG13, she made it a 100% match over 5% of the genome. So it's a bit of a bogus argument that she's come up with. In other words, Alina Chan's making it sound as if she's trying to divert people's attention away from her lab, but that's essentially what Xi Zhengli did. She brought attention to her lab when she published this thing. So Alina Chan went on to only, outside of her book, she only published 1 scientific paper. She's an MIT postdoc, and it was basically that SARS-CoV-2 was well adapted for humans. I kind of like to pick on this paper because it basically claims humans are the only mammals on the planet. Humans are mentioned 23 times. But mammals are not mentioned at all. SARS-CoV-2 was pre-adapted for mammals, of which we are one of them, many, on the planet. 5,000 out of total are humans. But that's essentially what she's claimed here. Humans are the only mammals on the planet. And she sees SARS-2 being, she's inferring, since it's circulating humans, it was designed for humans. It's a bit of a story, but SARS-CoV-2 was designed for bats. American lab bats, designed for Chinese lab bats. But you can tell that bats are quite the thing for virologists, because here in 2018, the Washington Post, which is the national newspaper here, was talking about them. And not just any kind of bat, the Egyptian fruit bat. You'll find that in the DARPA diffuse proposal. You'll find that to be the SARS-2 reservoir host. So that's a bit of a clue there. This is the CDC that would kidnap these Egyptian fruit bats from Africa. They're located in Egypt, of course, but also along the western African coastline. And they are here being harvested essentially from a mine in Uganda and brought back to the CDC lab in Atlanta, Georgia for trials. They're virologists, like the CDC and Ralph Barrick and Limfa Wang and even Xi Zhengli were fascinated with bats. And in particular, Western virologists are fascinated with their immune system. It's the immune system of the bat that's so intriguing to them, essentially. They are able to harvest these lethal viruses, without any side effects. It's just, you can just see them talk about bat immune systems all day long here. It is the obsession of bat virologists and bat immunologists. So here's another paper in 2018 about the batty explosive history of bats in the military. It even talks about, this is 2018, it talks about Mexican free-tailed bats being recruited as part of a bat bomb. But guess what SARS-2 infects Mexican free-tailed bats? Guess what was used as a proxy in the SARS-2 bat vaccine, or the DARPA diffuse grant, the Mexican free-tailed bat. So it's another clue. And then they also mentioned, of course, Egyptian fruit bats. It's no coincidence that SARS-2 infects Mexican free-tailed bats and transmits in Egyptian fruit bats. These are 2, lab rats being used in Western virology labs. And, bats are now the new frontier. They got a tank-like immune system, and so they're intriguing to these virologists who want to study them. So carrying on with the idea, this is kind of where another myth that formed, SARS-2 was designed for humans, because they often reference this paper here. And at the top of this paper, written by Petrosky, is the Homo sapien, us. And there's a few other: pangolin, dog, monkey, hamster. This one actually is pretty relevant. But the actual paper itself is irrelevant because it's in silico. That means computer simulation. In vitro would be somewhat relevant, but not really. That's in a test tube, a petri dish. The best one is called in vivo, in the living. That's actually animal-to-animal transmission. Ideally in the natural, but it's really only easy to test it in a laboratory. So that's what we want in vivo. But out of all these, the only one he really lists that's relevant is hamster, a Syrian hamster. That's an animal that's not found in China, but it is found in Western biolabs and DARPA preempt proposals. So this was the Petrosky paper. It's in silico. And a lot of people ask me about it, since they go with this idea that humans, are the targeted, species of the SARS-2 genome. And I'm like, no, you got to think outside that box. So this is a good in vivo paper here. I point people to, this is a European CDC paper, and it's pretty damn, thorough. It's talking about transmission models of SARS-CoV-2 in vivo. American mink, that's a clue. Those are not found in China, but they are found in Western biolabs. Raccoon dog, that is found in China. But it goes on to say there is no transmission of raccoon dogs found in the wild. There was no GISA'd genome for raccoon dog transmission ever uploaded. They found one outbreak, but it was next to a mink farm. So there's never been a recorded case of one raccoon dog infecting another, or an outbreak on a raccoon dog farm. The cat, they kind of go on to poke a hole in that. It's usually human to cat transmission, not cat to cat transmission. Ferret, they go on to poke a hole in that. It's about 25% efficient, not 100%. Hamster's a good one. Chinese, the house mouse, which is the lab mouse. That's not a great one. To claim that, SARS-CoV-2 infects house mouses, to claim it infects raccoon dogs, is the same infectious dose, 100,000 infectious particles, versus a hamster. It's only 5. 100,000 infectious particles shoved up the nose to get an infection? 5. Only 5 to get a hamster infected. Egyptian fruit bat, I show in the book. It's the natural reservoir host for SARS-CoV-2 with bat-to-bat transmission and the smoking gun email. They are not found, obviously, in China. They are found in Western biolabs, though. Deer mouse is another one. That's the American deer mouse. It's kind of the American rat of the Midwest. That eliminated all the labs on the Eastern Hemisphere in the U.S. and pretty much just the labs around the Rocky Mountain area, to be honest with you, because that's where these deer mouse are usually found. And white-tailed deer. That eliminated a bunch of labs. You got to have a huge animal lab in order to have a white-tailed deer experiment going on. You will not find American white-tailed deer, North American white-tailed deer in China. You will find muntjac and Sika deer. I think I got Angela Rasmussen to admit to me that muntjac are not susceptible, and no one's found anything in Sika deer. And there's also called a roe deer or red deer in Europe. It's not susceptible. The only deer are the North American white-tailed deer. It has a unique receptor binding domain, and they're also used for cell-spreading vaccine research by American researchers. So once the virus spilled into deer, it was pretty obvious it was not antibodies, it was virus. There's a big difference between antibodies like cats and dogs and virus. There was living virus in deer, lethal levels of virus. You're talking like, PCR values that would make your eyes, pop out. It was an insane amount of virus living in deer, live virus, without an immune response. So obviously COVID-19, if you think about it semi-logically, I know it's kind of hard to get your brain around it, but COVID-19, before emerging in Wuhan, had seen the snout of the North American white-tailed deer. So that's the clue that drove me, to really, push forward on the Rocky Mountain Lab origins for aerosolizing the agent. But I'm not going to get into that here. I want to stick to the genome and the myths around it. So this was another myth, per se, that formed around Shizheng Li and the Wuhan Institute of Virology. So if you look here at 652, what is it, 652? I don't even know where they stopped this thing. What does she claim, actually? I got to read what she's claiming here. Why does the alignment of the Nature paper stop right before the furin cleavage site? Why leaving this white space in the bottom right corner of this figure? All right, she's talking about a white space and whatnot. Essentially, the accusation is that she is hiding the furin cleavage site by cutting off the alignment at the S1, S2. But if you go into her previous work, this is a 2012 paper. She did the exact same thing. She always cuts it off right there in the bottom right corner. Here in 2017, she did the exact same thing. It's the 691 spot where she cuts it off. Why does she do that? Because that's where her software cuts it off. It's this old ancient software she uses based off MS-DOS, and it's 20 years old. And it cut the alignment at the exact same spot every single time. So this 2017 paper and this 2012 paper are the exact same thing of what's happening here, of what she's claiming to do. It's a bit inside baseball, but it was another theory that came about. So the paper, or the next step, the myth that formed was RATG13 is fake. And to claim that RATG13 is fake is to also claim that SARS-CoV-2 is fake. It's just, it's a bit preposterous. There's so much overlap between the two. It's to call one fake, you have to call the other fake. So to call RATG13 fake, you have to call SARS-2 fake. So that's doesn't really hold up unless you want to go down that path. What else do we have here? Give us a second to load. We talked about the slide of Barrett comparing SARS-2 to SARS-1, and he doesn't ever declare it. He's the one hiding the furin cleavage site. The genome, or the database going offline. So once you admit that it's biologically impossible to publish something closer than SARS-2, or to publish something closer than RATG13 to match up with SARS-2. We don't need to see the genome. But even so, the genome actually stayed online. The conspiracy is that it went offline in September of 2019. It actually was available until February 2020. So that's well after a lab leak. You had to have internal access to get into it, but it was still online. It's not offline. So, there's archives of it here in October, November, December, January, and February, where it finally went offline. That's well after a lab leak. So and even then, even if you do think that's a conspiracy, good luck trying to find a closer match, because I argue it's biologically impossible to publish something closer than RATG13. It is impossible. It's just not, really debatable. So, this is kind of getting into the weeds, but it's important. This is Ralph Barrick finally emerging from his bunker after 3 years of total silence and radio silence. After DARPA Diffuse leaked, he's come out to basically say what everyone else has said, including Christian Drosten. The WIV doesn't use my technology. They use WIV1, and it does not use type 2S restriction sites. So I think there's a sequence signature in that. And so he basically says they can't create a novel genome with my no-seum technology, because I use different kind of technology. And Stephen Goldstein, a coronavirus postdoc at the University of Utah, said the same. Barrick, this is basically a natural origin scientist arguing with a lab leak scientist that Barrick's lab was the one that proposed the creation of the SARS-like CoVs, and as did Xi's lab using Barrick's tech. They use different technology. Similar technology, Richard Ebright argues here. There are so many ways to assemble an infectious clone from fragments that the ends generate by type 2S restriction sites. Barrick just said they don't use type 2S restriction sites. I don't think they engineer with type 2S. So Richard Ebright's claiming he knows more than Ralph Barrick, which is kind of funny. And Richard Ebright's doing more to defend Ralph Barrick than Ralph Barrick. So Barrick did train them in seamless ligation, correct? No, because the WIV, Xizeng Li, uses bacterial artificial chromosome. It's extremely dissimilar from Barrick's technology, which is ligating 7 cDNA fragments, then in vitro transcribing and electroporating. So that's kind of a long story short there. That's kind of the way this whole thing's flipped. It's kind of funny that people think this, and I don't really understand why. But it's all answered here before DARPA Diffuse leak when Ralph Barrick was talking. Their collaboration is not a close one. She told me there was no exchange. This is Xizeng Li talking essentially now. Xizeng Li is now reflecting upon that collaboration of hers, and she didn't have the necessary tools to do the genetic kind of research, so she had to send these sequences to Ralph Barrick. So like SHC014, like WIV1, she also obviously sent RATG13 to Ralph Barrick sometime around 2018, 2019. And this, the collaboration is not a close one. There's no exchange of staff or anything. So, but they did exchange genome sequences. So this is, I'm trying to sort out the crazy in all this. It's called the division of labor more than it is called the, some sort of massive conspiratorial cover-up or something. Before the lab leak, this was a pretty good working relationship. But it's such a technical subject. Even the guys that wrote the great paper about finding those 5 restriction sites, 6 elements in the SARS-2 genome, they all 3 thought that the WIV used cDNA, and you just order in the mail. And no, they don't use cDNA. They is the virology. They use bacterial artificial chromosome. So it's just another myth that kind of took its own form. And this is Xizeng Li actually uploading the RATG13 genome before lab leak, which partial upload, but here it is. It's been named RATG13. Here by the time she uploaded it in July of 2018, she's doing a partial upload of it. So this is to the NCBI database, which had it in embargo until August of 2022. So that's why she had to go to GISAID to upload the genome, because it was embargoed on NCBI. So she went around. Xizeng Li, who I call the whistleblower, the hero of my book, he has gone around the NCBI, that's the NIH database that's embargoed it, and now she's gone around to the competitor NCBI to upload it. This is how crazy this has gotten. She had to go around the embargo to get this genome out to the public and send out the signal that this thing's engineered. So it's a total role reversal, really, in essence. The good guys become the bad guy in Ralph Barrick, and the bad girls become the good one in Xizeng Li, in my opinion. So this is a bit inside baseball here. I don't know if I want to get into it, but this is Richard Ebright. I will do it. This is Richard Ebright claiming that Xizeng Li has human airway epithelial cells, which was another lie, another myth perpetuated by yours truly.
Going on, we'll start with Dr. Ebright.
So, as you mentioned,
the Wuhan Institute of Virology constructed novel chimeric SARS-related coronaviruses that combine the spike gene of one coronavirus with the genetic information of another. They showed that the resulting viruses efficiently infected human airway cells and efficiently replicated in human airway cells. And they showed that the resulting viruses exhibited up to 10,000-fold enhancement of viral growth in lung.
So he said human airway cells. Xi Jinping uses vero cells, which delete the furin cleavage site immediately. I pointed that out to him, and he went on to concoct that kind of, cockamamie theory. In the paper he referenced to claim that, UNC was going to do the human airway epithelial cell research at the University of North Carolina, which is a U.S. government select agent lab. So that's another myth. There are no human airway epithelial cells. There's only vero cells, monkey cells, inside the WIV. HAE, human airway epithelial cells, don't delete the furin cleavage site. Vero cells do delete the furin cleavage site. I've covered that in another video. This is the paper that Richard Ebright will often reference, claiming that the Wuhan Institute of Virology and Xi Jinping could have engineered something like SARS-CoV-2. This is a 2017 paper publishing some novel work. Novel work means shuffling spikes into the WIV1 genome. That's all what that means. That's not really novel in the grand scheme of things. But you'll notice what they do use is vero cells, which delete the SARS-2 furin cleavage site immediately. It's a big issue. I've talked about it in other videos. I'm not going to go into it here. And I'm not going to pull it up, but in one of these links is a recovery map, basically showing how efficient they are creating a novel chimera, or at least recreating a novel spike in an existing backbone like WIV1, and they're only successful at 25% recovery rate. So this is where I get the, I hate to say it, but I'll say it. This is where I get the term Chinese junk from, because that's what this paper is, Chinese junk. A 25% recovery rate on a shuffled spike into an existing genome called WIV1. And it's not very efficient use of genetic engineering. It's better than most, if not anyone else in the research of coronaviruses, but they're still at the kind of the kiddie pool. So you'll often hear about someone in Xi Jinping's lab being the patient zero. That's a myth perpetuated by one man who we'll name here in this video. This is the other part of the book, what I start off with, that kind of punches people in the mouth. I say a $100,000 bet was lost because until now there was no biological evidence for one lab leak. So that $100,000 bet was this. This is the root claim debate between Pete down here and Yuri Deegan, a pretty prominent lab leaker up here. And so this was the opening of a 18, 20-hour, multi-day debate based mainly upon Bayesian analysis. But this was a good opener, I thought, by the natural origin Pete against the lab leaker Yuri, and these are the two judges here. So let's just watch.
So I wanted to start with a bit about the perception versus the reality of the Wuhan Institute of Virology. You read all these news stories, pictures of this being this huge building full of frightening viruses. I wanted to kind of humanize who we're talking about here.
That's Xi Jinping, that's Linfa Wang, that's Peng Zhu, that's Bin Hu.
Xi Jinping's group. She has a few scientists she works with, a bunch of grad students. These people on the right are who you're blaming if you think they created this virus. And I think the scale of their operation is probably smaller than what we're imagining here with this group that's creating all these chimeras, all these different furin cleavage sites. I found something in the diffuse grant. It looks like these are their salaries. Ben, who is frequently called patient zero, he's making $11 an hour. I'm not saying he can't, destroy the world at that rate, but I think we could solve biosafety pretty easily by just
Look at the smirk on Yuri's face.
giving him like a minimum wage job in the U.S. instead of whatever we're talking about in this.
And then they're talking about the consensus sequence. I don't want to play that, but essentially they're talking about Ralph Barick's consensus sequence. It's what he does, and I'll talk about that in another video. What are they talking about here? I don't want to talk about that one. And then they're talking about the HAS2 mice. This is pretty good, so I don't want to get too far into the weeds here, but people like Richard Ebright and tons of lab leakers claim SARS-2 infects humanized mice. But the virus that emerged in Wuhan does not infect humanized mice, BLT mice, HAS2 mice, whatever you want to call them. Never in the history of COVID, the most contagious disease on the planet, has one humanized mouse infected another.
On top of speculation.
I definitely what technology. Like inserting a furin cleavage site doesn't require.
No, but like the HAE cells, you go through and they've never used those before, so that's another step in there.
Well, I mean, using primary cultures, I think they actually used. Well, in any case, yeah, but using a cell culture is not like a technology. And they definitely had humanized mice. Barick sent the humanized mice to Wuhan in like 20
Yeah,
whatever.
but those don't work because of the 501Y mutation. That wouldn't make COVID. So you're really restricted by. That's why I looked into.
501Y is like a 2021 mutation that can infect mice, but that's 2 years after the ancestral strain emerged.
all these culturing experiments, like how could they make it? And also like if it did make, if you did use the mice, it could leak. It would just be a different virus because it would have the 501Y mutation.
Yeah,
So if they did that first,
it
and
necessarily would have absolutely had the 501 if it leaked from mice.
it's like
Again, to claim SARS-2 infects mice is to also claim it infects raccoon dogs. It's the same dose, 100,000 infectious particles shoved up the nose of one animal, and it does not transmit to the next.
the second generation mutation. Like it happens really fast in the serial passaging. So, it could leak in the first generation. And maybe that's even a more likely way for it to leak because like we have these leaks from Taiwan, right, where it actually got out of SARS-2 got out of the lab, but they were like working with mice each time. And they, yeah.
Maybe,
So
Later variants.
I was going to ask a different question, but let's go to this then. Okay, so you talked several times about how you don't think that SARS-2 could have been cultured in a specific way because when people did that actual experiment, they observed specific mutations which are not present in SARS-CoV-2. And to me, it's not obvious that this is consistent and replicable. But now you're saying if you attempt a passage.
I think they're just going to keep talking about the same thing. So that's this is where we'll end it. This is at least this root claim debate.
started by Steve Bannon
turned into a
in
bit of a
January 2020, and the Sky Miles Glow.
root canal.
They worked with Li Meng to make the early theories. They went on Fox News with it.
This is where a lot of the early theories came from. Bioweapon, PLA. There's no PLA. There's no CCP inside the Wuhan lab before 2020. Done. No one's found one. Not one member. It's a lab built upon the idea of collaboration with the West. There's Danielle Anderson in the BSL-4. That's pretty much it. And she's got it more or less to herself.
They said it was a bioweapon. They all went on to say the 2020 election was stolen. Both these guys were later had some legal trouble. The Miles Guo was defrauded people out of $1 billion when they searched his apartment. His apartment building was lit on fire. 2021, this kind of morphed into a new version of the lab leak theory. And it was really spread by this article in Wall Street Journal by Michael Gordon. That came from a Trump State Department memo first. And it came from this guy, David Asher, who says
I name him in the book.
the three got sick. Sometimes he says November, sometimes he says October. Sometimes he says a monkey bite at the lab that caused this accident. Sometimes he says one of their spouses died. And
So that's where David Asher got the rumor from two foreign scientists working in Wuhan. He did that on a podcast. So it was an online rumor that he perpetuated, and he misspelled one of the three names. I think we get into that later.
And the Wuhan scientists were acting normally this whole time. They were traveling to conferences. They were going out to dinner. And the guy who spread this...
And, young postdocs don't get sick from COVID. They just don't. Old people, yes. Sick people, yes. But young, healthy postdocs just don't. So trying to find evidence of a lab leak from a young, sick postdoc is a bit of a, misnomer. Danielle Anderson says she wasn't sick, and I believe her. I mean, this thing doesn't, if you're young and healthy, you probably never got COVID. If you did, you didn't know it.
In the Wall Street Journal is the guy who announced that Iraq has weapons of mass destruction.
That's the guy that published the David Asher rumor, the weapons of mass destruction guy, Michael Gordon of the Wall Street Journal.
He spread multiple articles about this. Every time he spread one of those articles, there's a huge spike in Google searches the same day as this article, because other articles reported the same thing with the Wall Street Journal as the source. This changed in 2023 to be 3 scientists at the lab with these names. The guy who reported that also says the U.S. has alien spacecraft a week before that, and then it got amplified by the aluminum tubes, Iraq WMD's guy. There are a lot of reasons to doubt that those 3 people were sick. I've gone through some of these, other than just the people that are spreading it, and the rest of the government intelligence can't confirm this.
So this was the declassified intel. It's kind of hard to read here, but I have it in the book. We are unable to confirm if any handled. Yeah, they're basically saying, it was nothing. Mildly ill, range of symptoms. Allergies is what they're talking about. So, it was a big to-do about nothing.
This looks like a misinformation campaign by other people who are involved with other misinformation campaigns who can't keep their story straight. It's highly politicized, but the Juana mark is the most likely place for natural origin. There are lots of lab leak theories. Most of them must be...
They, will diverge there from Pete's, natural origin market, because they can't answer the question why Wuhan. They never have, they never will. Why the Wuhan wet market? No, why Wuhan? I don't want to talk about the wet market, just why Wuhan. So that they want to pull you into that Wuhan wet market debate. You stay away from it and you just ask why Wuhan, and they can't. But DARPA Diffuse can't answer it. There's another question here that Yuri Degan can't answer, and that's the publication of RATG 13. He goes on to say, in fact, I think that's why she published RATG 13, so that they would shut their stinky mouth, the artists of the HIV paper. The HIV paper inserts another misnomer, myth. Those HIV inserts are also found in RATG 13 and Nospinol 52, so they aren't really inserts. They're just existing, pieces of evidence that were already in consensus sequence-like genomes that could be used to create something like SARS-CoV-2. But Yuri Degan doesn't hold up, because the HIV paper was January 31st. She's only uploaded on January 24th, a week before. But he was well ahead of his time. There's no way to take that away from him. And here's Yuri Degan in April of 2020. Read it. It'll make your head hurt because he gets into such detailed analysis on the genomic engineering. It's an icky subject. There's no way around it. Live bat research is even more icky than genomic engineering. But, he goes on to say, he talks a lot about RATG13, so it was always on his mind about why the heck did Xi Jinping even publish it. And he noticed that it was published on January 23rd. So we're going to talk about someone that lied about that publication date, which is a bit crazy, but they did. But Yuri, he just deviated at one point that I would, try to drive home. He claims that Barrick is a competitor of Xi Jinping. And he's really not, because Xi Jinping shares samples with him. But then he'll claim that, Xi Jinping is on par with Ralph Barrick, and she's really not, because she uses bacteria artificial chromosome. So, this is a good picture. Here they are. That's Ralph Barrick. That's Xi Jinping. There's Linfa Wang. There's Danielle Anderson. This is October 2018. This is the DARPA Diffuse proposal, but it's being funded under Tony Fauci's NIAID, because Peter Daszak right there found a way to get everyone's name back on the NIAID grant. The R01 grant essentially became the TA1 in DARPA Diffuse. I'm not going to get into that here. I've done that with other videos. But this October 2018 picture was pretty telling. So a lot of evidence, though, that Yuri used was based upon this 2015 paper. He's making the claim that Xi Jinping went ahead with something like DARPA Diffuse on her own, and he'll if you ask someone like Yuri Deegan or Neil Bleeker, provide one paper showing a Chinese scientist with interest in a furin cleavage site in a novel coronavirus, and they'll usually reference this paper. But it's the University of Minnesota. First and last article always show the lab it was done at. Ralph Barrick's on there, but so is Xi Jinping. She's on there for sharing the bat cells. So that's what she did. She shared the bat cells. But the paper is talking about proteasis, which is essentially one of five is furin. So they are showing interest in a furin cleavage site. And Ralph Barrick told us why, I believe, here in this video. And it's not a video. It's a TWIB podcast in 2015 where Ralph Barrick's talking about that paper we just looked at. So it lets you know who's the writer of the paper.
Entry and cross-species transmission is proteolytic cleavage of that.
He was talking about the RBD.
The second major barrier for...
The first was the RBD. The second's going to be
coronavirus,
proteasis, furin cleavage site.
Entry and cross-species transmission is proteolytic cleavage of that spike. And so the coronavirus spike is generally broken into two pieces, an S1 N-terminal domain of about 90 kilodaltons and a C-terminal 90 kilodalton domain. Proteases, and there's 4 or 5 depending on who's doing the study, different proteases have to cleave that spike to cause conformational rearrangements to expose a fusionogenic domain, and that fusionogenic domain then fuses the envelope of the virus to the surface of the cell, or the surface of an intracellular vesicle to mediate entry into that cell. In the case of coronaviruses, there are proteases that can mediate that event at the surface of the cell and intracellularly. And so depending on the spike, the cleavage events may occur on the surface to mediate fusion to the plasma membrane or fusion to internal vesicles internally with the intracellular proteases. Go ahead, Rick.
So those are host proteases.
He's talking about intracellular proteases. He's talking about how to cross-species transmission occurs from a bat to a civet to a human, or straight from a bat to a human. And he's noticed that the furin cleavage site can get the bat virus into the bat cell and cause that to happen. Now he's talking about bat cells, which are very similar to human cells, unfortunately.
And this proteolysis happens in the process of entry, not before, like flu. Okay, great. I got it.
So,
thanks.
For example, in collaboration with a researcher at University of Minnesota named Fang Li.
Now he's talking about this paper. If I can get back up to it. Fang Li, University of Minnesota. I'm proving the point. This isn't Xi Jinping's research. It's Ralph Barrick's.
Fang Li has shown that HKU4, which is a relative to MERS coronavirus, that HKU4 strain can bind to the same human receptor that MERS does, which is dipeptidylpeptidase 4, and it binds very well to the human DPP4 molecule. However, the barrier for entry is not in receptor binding. The human proteases can't cleave the S2 spike and can't activate the fusion domain. So the major barrier for HKU4, and the reason it's not infecting human cells, is because of proteases. Host proteases not being able to process the spike appropriately.
So this is a 2015 interview with Ralph Barrick, and he's already zeroed in on a furin cleavage site. So he's obsessed with it, to be honest with you. Once you know what to look for, there's a list of papers down here where you can see him focusing on it. Here's a 2018 paper where he wants furin, essentially, at the S1S2. There's another paper here in 2017 where he's talking about proteasis. In 2023, after the pandemic, he's still talking about it. It would be a furin cleavage site at the S1S2 border would would be interesting, but urge constraint on doing that kind of research. I mean, Barrick's he's utterly obsessed with the furin cleavage site, and here he is in 2019. We'll talk about some of those papers here. This is the 2017 paper. This is, Barrick turning into a bat immunologist, essentially, jumping species, a mechanism for coronavirus. He's talking about spike binding and cleavage necessary. He's talking about the proteases sites and that can cause that. And then just to drive home the point that he's turning into a bat immunologist, here he is. He talks about the ORFs, the open reading frames, in the genomes of coronaviruses, and then he's also referencing lymphoweighing, 4 or 5 times. So that's Ralph Barrick in 2017, turning into a bat immunologist, bat virologist of sorts. Here he is in 2018. This is an interesting paper, lysosomal proteasis. He just straight up says furin in this paper. They're infecting bat cells with the furin cleavage site, and they do this again at Feng Li's animal lab. And I believe, Xi Li's not on here, but they are doing the Chinese horseshoe bats just to show you how obsessed they are with the Chinese horseshoe bats. They're infecting them with furin cleavage sites. And so here's another paper without Barrick, without Xi Li, and here he is basically declaring trypsin unlocks the barrier. So he later wanted to testify that it's a nice, cheap, easy way to get a synthetic Chinese genome to grow my North Carolina lab. So here he talks about the furin cleavage site essentially. He's talking about MERS here, which is a work around the SARS select agent list, but RXXR, the furin cleavage site is RRAR and SARS-2. So he's basically publishing this thing, and it gets even crazier than that because Ralph Barrick in that paper referenced the precise molecular blueprint for SARS-CoV-2. I didn't even include this in the book, but it is here in the Substack. This was found by the German scientist Andreas. This is a crazy find, but I believe it's reference 41 down at the bottom. There it is right there. Let me go back to that. So similarly, this is Ralph Barrick in 2019 talking about this mouse-adapted virus MA30 that can alter proteases activation and entry in vivo. And so what is he referencing? He's referencing this Stanley Perlman 2017 University of Iowa paper, which had the precise molecular clone for SARS-CoV-2. What is it? It's called PAT7, molecular address. Don't want to get too far into the weeds here. PRRVRSV is what it is in 2017. And the SARS-2 furin cleavage site is PRRAR. A lot of times from natural origin scientists, what about the proline? Why is it there? Well, that's why. It's a PAT7 reference that Barrick did himself in 2019. So I asked Perlman, what the heck is this, why is this molecular clone being referenced in a 2019 Barrick that's found in the SARS-2 furin cleavage site? He never responded to me. He did respond, Stanley Perlman, to my book survey. I mean, he would communicate with me. Now he's no longer. So we'll finish up here. It's going a bit long, but these are more myths basically around the Ralph Barrick, idea. I just want to drive home the fact that, DARPA diffuse was his baby and his idea was to infect bats, live bats, Chinese horseshoe bats, cross-reference with TLR3 and TLR4 to your human body. That's kind of a nasty little thing right there. So that's basically something Ralph Barrick's talking about, DARPA diffuse, that's found in SARS-2. So you'll find out what it is. But yeah, a lot of people fight me on, he's not talking about a bat vaccine. That's literally what, these are Ralph Barrick's words, BR. He's talking about, virus burden is bat, reducing disease risk in these animals for years by creating these novel chimeras. So this is Peter Daszak to Ralph. I clearly don't understand the details of your approach. And Barrick goes on to this crazy, long, detailed idea that I'm going to zap bats with live coronaviruses and try and upregulate their immune system. So it's on. Call it what you want. It's a furin cleavage site, inside of a novel coronavirus to get it inside the bat cells and create a bat vaccine. So why is he doing this? This is Ralph Barrick. There's about 90,000 U.S. troops in the Southeast Asia area, mostly Japan and South Korea. It was a military grant. This is the DARPA diffuse proposal draft notes that came out by U.S. Right to Know in early 2024. So we got to see a lot more detail. The brilliant U.S. Right to Know scientist or journalist went to the USGS to get them. Ralph Barrick won't give us this stuff, but she went to the USGS to do it. It's a bit of a FOIA move. It was a brilliant move. But here's Jonathan Epstein. He's basically Peter Daszak's number 2. He may be running EcoHealth now. I recorded this in 2025, and I don't think Daszak's no longer affiliated with EcoHealth, but Epstein is. And this is John Epstein of EcoHealth. If we're proposing experimental work with bats, we should specify that we'll use SARS and SADS of pig diarrhea, Chinese horseshoe bats, which we can readily obtain by our Chinese colleagues at the WIV. So this is why Wuhan's a story. That's it. In 2018, EcoHealth, Ralph Baric are talking about experimenting with bats at the Wuhan Institute of Virology. And you had to go to Wuhan to experiment on the Chinese horseshoe bats, which is the source of SARS-1 and SADS, which is this pig virus that jumped all the way over here to America, for a few times. But I think the first was 2013 or 2014, and it wiped out like 25,000 U.S. pigs. So we're an interconnected world, and it came over on pig feed. I think it's a weird story, but SADS is it's a sister virus to SARS, and it infected American pigs, but it came from Chinese bats. So you had to go all the way to Wuhan to test your vaccine on bats, the Chinese bats that harbor these viruses. So that's the reason why. And this is Ralph Baric saying, vaccination is a positive, provides long-term immunity. Let's not attack one arm with the other. So Baric is pro-vaccine going into this thing, even though it doesn't call it a vaccine. They're just throwing the kitchen sink at this thing. So this is, Baric here is talking about sending, I don't know, I think he's talking to Linfa Wang. We can provide you SHC014, which we can provide. Baric can send the SHC014 genome to Linfa for testing on live bats, is what he's saying there. So that's the DARPA Diffuse notes without getting too far into the weeds. I'll upload all these links to the website. There's one more thing that I wanted to end on here, and it's another myth. Here it is. A lot of this you'll hear about, this comment right here is talked about ad nauseum. PD, Peter Daszak, he's talking to Ralph Baric, and she's saying, if we win this contract and impose all this work will necessarily be done by Ralph, but I don't stress the U.S. side of the proposal. Once we get the funds, we can then allocate them. I believe some of these assays can be done in Wuhan as well. Assays is a PCR test. And so here in the draft, they say conduct binding assays in humanized 2 under Ralph Baric and humanized mice. And he's proposing that we can do this in Wuhan. Well, guess what happened in the final draft? It's right there, binding assays in humanized mouse work under Dr. Xi's lab. PCR testing and humanized mice have nothing to do with SARS-CoV-2. Okay, so that's that. This is what it's about. Deep in the DARPA Diffuse proposal, Ralph Baric wants to test a novel chimera called HKU3S mix. It's about 25% different from SARS-2 or SARS-1, and it's a human codon optimized in vivo testing in the living in bats. Baric wants to test his patented chimera HKU3S mix. I'm not going to get into it here, but that's SARS-CoV-2 in bats in Wuhan. That's the name of the game. That's what it is. So that's all of DARPA Diffuse in a nutshell, and that's how Baric's genome wound up in Wuhan. And so once you understand that Fauci funded DARPA Diffuse and his genome leaked from the Wuhan Institute of Virology, Baric's genome, a lot more makes sense. When Baric finally showed up after 3 years of silence, he gave up, the simple fact that he was on the February 1st call, and no one thought that. No one, absolutely no one, because it wasn't anywhere in the records, but Ralph Baric thought it was a public record that he was a part of this February 1st teleconference, so he just gave up something that he didn't know was public record. It was funny. And it came up at the beginning. This is line 327, when they go around and ask, have you talked to this? Have you talked to that? Have you talked to Fauci? Yeah, I talked to Fauci. I was on the phone conference with him on February 1st. It had to do with the origins. So Ralph Baric was secretly listening to the February 1st teleconference, and Tony Fauci invited him. So after Tony Fauci was told, that's how I open the book up in the first few pages. After Tony Fauci's told SARS-CoV-2 has been engineered, instead of calling the FBI, he called Ralph Baric. And so this is Bryce Nichols, who's a colleague of our buddy Richard Ebright. And he's pointing this out. It's pretty obvious, isn't it? And here comes who. Different call. He's on the call, February 1st call. He's claiming it's a different call. The different call would be the February 3rd call, where Ralph Baric attacked Christian Anderson. So Ralph Baric has heard Christian Anderson's evidence on the February 1st call and ambushed him on the February 3rd call. But, for whatever reason, Richard Ebright wants to protect him. I'll come to your own conclusions as to why, but I think it's pretty obvious who's covering this up. It's not even, Ralph Baric. So we'll end it here on a semi-high note. I've been called a second-generation lab leaker. And I don't want to drag him into this. He doesn't have much to do with this, but he came along even after me, well after me. And I think we call him a third generation lab leaker. He just read DARPA Diffuse and read the proximal origin paper. He is Bryce Nichols, who's again a professor at Rutgers University, along with Richard Ebright. And here he is, or here they all are at a conference. And watch what happens.
Pattern in SARS-2 with the high concentration of silent mutations is with enzymes. These two enzymes have only been used together on a coronavirus infectious clone once before, pre-COVID, and that was by this gentleman Ben Hu and colleagues at the Wuhan Institute of Virology. And so there's some kind of ways in which there's a concealing lab origin theory revolving around this grant. That doesn't mean everyone who wrote it acted on it, but with that grant circulating, if you gave me that grant in 2018, and I used that to predict the emergence of SARS-2, it would be much more accurate prediction than using the methods of the time for wildlife biogeography and viral emergence.
Can I jump in on that diffuse proposal for a second to say it's very important that we discuss where the research was actually proposed to do, because the two scenarios have been laid out as research in Wuhan or natural. However, the research proposed to insert furin-cleavage included Ralph Barrick at UNC, and in fact, the work was proposed to be done at UNC, and in fact, my understanding, I believe, is that we don't have the full grant proposal. It hasn't been released, do we? Correct? The US government has not released the full grant proposal, which would include details that might help us know where that research was actually really proposed to have been done. We are basing the idea that it was done in Wuhan because there's a footnote in a draft that says we can offload, well, I'm paraphrasing it, we can do this research in less biosafety levels in Wuhan.
Yeah, they go on to talk about, a sideshow. You just got to focus on the main show. In the DARPA diffuse proposal, Ralph Barrick will lead the targeted immune boosting work extended to a small group of wild-caught bats at the Wuhan Institute of Virology. That's it. That's the whole name of the DARPA diffuse game. That's what Barrick wants to do. He wants to test his novel chimeras, not in his mice. You don't get $14 million from the military or Tony Fauci to infect humanized mice. But you could do it to play with bats, which was the frontier of virology in 2019. And that's what he wants to do, test his novel chimera in bats. HKU3S mix again is the SARS-CoV-2 chimera. That's what it's all about. And a lot of people get sidetracked on these other things. So the SARS-2 genome's in the DARPA diffuse documents, the plan for Ralph Barrick to test this in the SARS-2 documents, and there's really only one person covering it up. I call Richard Ebright the unwitting participant, which is essentially a good, the same time Christian Anderson wanted to call the FBI. Richard Ebright's telling the Washington Post there's absolutely no evidence of genetic engineering. He's telling Chinese state media no evidence of genetic engineering. At the same time, everyone else wanted to call the FBI and the CIA. So we'll end it there. This ran a bit long, but I'll just finish off down here on paragraphs 3, 4, and 5. Ultimately, Ralph Barrick's furin cleavage site was the gateway to a much bigger story. By November of 2022, I notified Richard Ebright that COVID-19 was a self-disseminating bat vaccine developed in Fauci's Biodefense Lab. From a February question to a November, declaration, that's how easy this was to solve. It's not hard. All you have to do is get over the hump and say Barrick instead of the furin cleavage site. And then once you do that, you realize he didn't, aerosolize the agent as Fauci's lab did. And, just to keep it simple, bats aren't birds. They're mammals like you and me. This wasn't nefarious. It was an accident. It wasn't some grand cover-up or grand conspiracy. About 5 people knew what happened. And even the 5 people came forward and said they did it, I'll be the only one that believe them. But my 446-page book barely mentions Ebright. But the premise was built around a single question. Why does this old world virus called SARS-2 love New World lab animals? Over 100, I asked nearly 300, were asked the same question, but my book provided an answer. A smoking gun email from a Colorado lab confirms that COVID was created in a Montana lab but tested in Wuhan. The case for U.S. lab origin was based on a fact check by Macer Joseph Murphy's letter to the Department of Defense. COVID infects U.S. lab bats, and its disclosure of the losing DARPA diffuse proposal, which referenced the same U.S. lab bats. Fauci's Montana lab won the 2018 DARPA project, and by 2019 he merged the project's winners and losers under his CREED initiative. So that's the origins of SARS-CoV-2, and that's how it came about, and most of the evidence was buried in our own backyard, as I say in the back of the book. So we just had to know where to look. So if you'll have any questions, feel free to contact me, jim.haslam@protonmail.com. It's a complicated subject, but once you get over the hump and see, the supply chain, it's pretty easy to follow. And it's got a pretty intriguing, interesting ending. So we'll end it there. Thanks, guys. Bye.